The science of prokaryotic replication. In prokaryotes, the DNA exists in naked form, unenclosed by any cell organelles. Bacteria have single circular genomic DNA that is present in cytoplasm. History. DNA replication is the biological process by which a cell duplicates its DNA, producing two identical copies from a single DNA molecule. This ensures that genetic information is accurately transmitted during cell division. James Watson and Francis Crick described the double helix structure of DNA was discovered in 1953. In 1958, Michelson and Stahl demonstrated the semi-conservative nature of DNA replication, proving that each daughter DNA molecule contains one parental and one newly synthesized strand. The DNA replication in prokaryotes require following proteins. DNA-A binds to origin of replication, ORI-C. DNA-C is a helicase loader. DNA-B, helicase, unwinds the DNA double helix. Single-strand binding, SSB. Proteins stabilize single-stranded DNA. Primase, DNA-G, synthesizes short RNA primers. DNA polymerase III is main enzyme that extends new DNA strands. DNA polymerase I removes RNA primers and fills in the gaps. DNA ligase seals nicks between Okazaki fragments. Topoisomerase, gyrase in prokaryotes, relieves supercoiling ahead by cutting and rejoining DNA. Sliding clamp, beta clamp in prokaryotes, holds DNA polymerase onto the DNA template. Clamp loader, gamma complex in prokaryotes, loads the sliding clamp onto DNA. Replication can be divided into three steps: initiation, elongation, termination. Initiation. Initiation begins at a specific site known as the origin of replication, ORIC. In E. coli and most prokaryotes, there is a single ORIC on the chromosome. The ORIC, origin of chromosome, is a specific site in E. coli where DNA replication initiates. Located at the 84.5 megabase pair unit position of the E. coli genome, it is positioned opposite to the termination site. The structure of ORIC consists of two distinct regions, the 9-mer and the 13-mer. The 9-mer is a repeating sequence of 9 nucleotides and is also known as the DNA-A box. The 13-mer, on the other hand, is a repeating sequence of 13 nucleotides and is characterized by a high proportion of adenine thymine AT base pairs. The 9-mer, or the DNA-A box, plays a crucial role in the initiation of replication. It serves as the binding site for the DNA-A protein, also known as DNA-A, helicase loading. DNA-A is a AAA plus ATPase that forms oligomers and breaks down ATP at a slow rate. It recognizes origin and binds to it. DNA-ATP hydrolysis allows loading of DNA-B-DNA-C complex. Here, DNA-A acts as an anchor which allows helicase, DNA-B, to load. Once DNA-B loads, it immediately opens DNA double helix and opens up the structure. The replication fork formation. Helicase continued to open the double helix, and at the same time topoisomerase enzyme rotate the helix and remove the strain that is caused by helicase. The opened-up DNA structure allows DNA polymerase to bind and start replication. The single-strand binding is prevented by the binding of SSB proteins. They also stabilize replication fork structure. DNA polymerase, clamp loader assembly, then completed, and initiation ends there. Elongation. There are 3 types of DNA polymerase that are involved in prokaryotic replication, in which DNA polymerase III is the main enzyme for polymerization. DNA polymerase III is the most efficient enzyme here, which is loaded in form of clamp-loader complex. It is an enzyme with multiple subunits. The clamp-loading complex of DNA polymerase III, consisting of parts of the two TAU subunits along with the gamma, delta, and delta prime subunits, is also an AAA plus A TPASE. Together, ATP and the novel B-sliding clamp form a stable combination. The dimeric clamp undergoes strain as a result of the binding, and the ring opens up at one of the subunit interfaces. The structure of DNA polymerase resembles palm of hand, where one strand goes inside the hand while other goes outside. This allows constant uncoiling and opening of helix and polymerization at high speed. DNA polymerization require DATP, DTTP, DGTP, and DCTP. Polymerase adds one DNTP at 3-OH and forms phosphodiester bond. It is a self-sustained reaction where energy is provided by breakdown of DNTP into DNMP and pyrophosphate. Leading strand synthesis is straightforward. Primase synthesizes first few oligonucleotides which provide 3-OH group for polymerase. It is called primer. Then DNA polymerase is pushed forward as clamp loader, and it starts synthesizing leading strand in continuous manner. Lagging strand synthesis is similar to lagging strand. Primase synthesizes primer, and DNA polymerase polymerizes based on template. However, since it is in reverse direction, the DNA polymerization is discontinuous, and only short fragment is synthesized, which are called Okazaki fragments. Lagging strands synthesis is tricky. Leading strand has 3-OH, thus polymerization is straightforward and possible. However, in lagging strand there are 5-OH group, so polymerization is not possible. Polymerase solves this problem by looping the lagging strand. Thus 3-OH group is exposed and polymerization can be possible. DNA ligation occurs in 3 steps. DNA ligase, I, is a cellular enzyme that catalyzes the covalent ligation of adenosine monophosphate to DNA via hydrolysis of adenosine triphosphate. Following a nick in duplex DNA, the ligase polypeptide transfers an AMP group to the nick's 5' phosphate termini. In a process involving nucleophilic assault by the 3' HO group and release of AMP, the non-adenylated ligase catalyzes the creation of the phosphodiester link. Termination. DNA polymerization in bacteria can be terminated naturally as polymerase can loop back and both leading and lagging strand encounter each other. The circular DNA replication split in 2. The 2 halves of an E. coli chromosome join at a terminus region. The terminus region is called TER, which contains multiple copies of a 20 base pair sequence. The TER sequences on the chromosome are organized in a way that traps a replication fork inside. Protein TUS binds to specific sequences in the tRNA terminus utilization substance. Thus, replication fork can be stopped by the TUS-TER complex. Each replication cycle only allows one TUS-TER complex to work, and that is the complex that is encountered first by one of the 2 replication forks. TER sequences may prevent over-replication by one fork in the case that the other is delayed or halted by encountering DNA damage or another impediment. Once TUS-TER complex forms, DNA polymerase gets disassembled. The finished chromosomes are united as catenanes, or topologically interlinked circles, as a result of replication of the DNA separating opposing replication forks. The circles are not chemically bonded to one another, but they are impossible to disentangle, without the help of enzymes called topoisomerases. DNA topoisomerase 4, a type 2 topoisomerase in E. coli, is responsible for transiently breaking both DNA strands of one chromosome and allowing the other chromosome to pass through the breach, therefore separating catenated chromosomes. Replication in eukaryotes will be available next week. The content has been taken from Molecular Biology of the Gene by Watson. Thank you for watching and subscribing to support free education.